So it is great to have Laura Volpicelli-Daley with us today. Welcome, Laura.
Thank you.
As you're probably aware, the way we like to sort of run this is I like to find out a little bit about you, then a little bit about your research, and then we can discuss, where your research is now, where you think it's going, and then what it means for the wider Parkinson's community. So, you're based in America, is that right, Laura?
Oh, sorry, you froze. I'm based in Birmingham, Alabama, at the University of Alabama in Birmingham. So
okay, and your life began in Birmingham, Alabama, or how did
no,
you?
my life began in Baltimore, Maryland. I've lived in many places in my life. My parents, they are from Philadelphia originally. They got tired of the crowds and the weather in the Northeast. So when I was a kid, they moved out to New Mexico. That's where I grew up.
So New Mexico, for people who live in the UK, that's quite a long way south, because it sounds as though it's near Mexico.
Southwest, yeah. Yeah.
And so during that period of time, did you go to university down in that part of the world, or?
No. So growing up, I didn't have, the best. I did not love high school and middle school. So I wanted to get out of New Mexico as fast as possible. Now that I go back, it's a beautiful state. I highly recommend anybody visiting. But I went to college in Pennsylvania, a small liberal arts school.
And when did you start to really get into sort of science? When did that?
So it wasn't until, I was a math major, English literature. I studied English literature in college.
Math and English literature in the UK are a pretty odd combo.
Yeah, it is. It is. Actually, the English literature was my favorite, but I only got B's. I'm not the best writer. My writing was dry and boring. So, but I would still go back and major in English literature again. So Thomas Hardy was my favorite author at the time.
He's not very cheery.
Yeah.
He's not a cheery chap, Thomas Hardy, I have to say.
No, but a realistic, very realistic.
Very realistic, yeah. And wasn't very nice to his wife. But anyway, that's a separate discussion. So having done English literature and loved it, you then decided to move into science.
So I started off working in addiction research center in West Philadelphia, working with patients. And that's where I became interested in the pharmacology. What drives people to addiction.
So this is drug addiction or alcohol addiction?
It was drug addiction, alcohol and opiates.
Yeah.
And so I got interested in that. And so then from there, I applied to graduate school. Okay. And I ended up in kind of a pharmacology lab looking at acetylcholine and Alzheimer's disease.
Okay. So this is a chemical which is sort of intimately linked to Alzheimer's and what the drugs largely work on.
Right. I mean, acetylcholine is really involved in cognition, no question. So, and I had a phenomenal mentor. So this was at Emory University. So Dr. Alan Levy was my mentor. And I think that's what really got me, stuck in science, just having him as my mentor.
And so you were working on acetylcholine and Alzheimer's, and how did you move into the world of Parkinson's? When did that transition take place?
Sure.
And how did that come about?
So it wasn't until I started to work at the University of Pennsylvania as a senior scientist with Dr. Virginia Lee and Dr. John Trojanowski. And so they, put me on a Parkinson's disease project. There are, Alzheimer's and Parkinson's are intertwined. I mean, so a lot of the same neurochemicals are involved. They share pathologies as well. So I got involved in a Parkinson's project, and there I was involved in helping understand kind of the prion hypothesis of Parkinson's. So in no way do I think Parkinson's is a prion disease, but a lot of these protein aggregates in a Parkinson's brain can go from neuron to neuron where they initiate pathology in a neighboring neuron. So that's kind of how I got into. So obviously that lab's very famous. They've done the most amazing amount of work. Yeah.
And so your project, you were on Alzheimer's there, and then they moved you to a Parkinson's project.
I started on Parkinson's there.
You started on Parkinson's.
Yeah.
And we'll come on to this, Brian, because this has obviously gained a great deal of traction and interest, and some of our previous Research Spotlight interviews have been, we've been interested in their work on this, but this is all related to this protein alpha-synuclein. So alpha-synuclein, it might be useful just to know a little. So alpha-synuclein is a protein we find in our brain, and it's quite a prominent protein in our brain?
Very abundant in our brain. And it's a normal protein. It's in all of our neurons.
Okay, so it's in every single nerve cell. And then in Parkinson's, it goes a bit wrong?
In Parkinson's, it goes a bit wrong. So normally, synuclein is probably bound to synaptic vesicles in the brain. Synaptic vesicles.
These are the things that release the transmitter from the nerve.
The chemical.
So the nerve ending releases dopamine, and those are in little vesicles.
Dopamine, acetylcholine, glutamate. Yeah. So all involved in neuronal communication.
Okay. So basically, a nerve cell will work by releasing these little packages, these little vesicles of transmitter. And you're saying alpha-synuclein helps regulate that process.
Helps regulate that, yeah. And then in Parkinson's, it stops binding to these vesicles, and then in the neuron, it'll start to form these abnormal aggregates. It just kind of build and build and build, and then spread throughout the entire neuron.
Okay, so all of us have alpha-synuclein in our brain,
right? And
it normally is there for providing some vital function to how we normally. And then in Parkinson's, for some reason, it starts to go a bit wrong. And dare I ask, what's the evidence for that? I mean, we can all have ideas on it. So what would be, because obviously, you've done a lot of work on this. What was the sort of first bits of evidence that suggested that this protein in particular had something to do with Parkinson's?
Sure. So there's a lot of evidence that synuclein is involved in Parkinson's. So Maria Spillantini and originally found, actually, let me back way up to the early 20th century. So Fritz Heinrich Lewy actually found Lewy bodies in Parkinson's brains. It wasn't really known what the protein composition of what these Lewy bodies were. It just, we knew that it was involved in Parkinson's.
So these are pathological hallmarks, Bugsy. You find these bodies inside the nerve cells.
You find it in the nerve cells, in the cell body. They're kind of, these kind of, kind of spherical blobs in the nerve cells, but then you also get kind of these thin neurites that are in axons, which again are important for the nerve function. So in 1997, Maria Spillantini found that synuclein is a major component of these Lewy bodies. At the same time, it was found that mutations in synuclein are also linked to a rare form of Parkinson's disease. So synuclein mutations are very rare, but they do cause Parkinson's disease. So that's what...
it wasn't just the preserve of a sort of rare genetic form of it. It was found in Lewy bodies, which are found in the brains of
most
Parkinson's, as well as, I guess, as you get a bit older, you probably just form a few of these anyway.
Sure. Yeah. So, and then there's other genetic studies that have come out showing that gene duplications and triplications of synuclein also cause Parkinson's disease. I'm sure people are familiar with genome-wide association studies. So just small changes in your DNA. They look at thousands of people and link small nucleotide changes in your DNA to Parkinson's disease. And synuclein is always in these genome-wide association studies. This is a strong hit.
So so there's a genetic link, both in terms of the population and in terms of these rare families, and then pathologically.
Exactly.
It's there. So alpha-synuclein for some reason doesn't like these vesicles in people with Parkinson's, and it starts to aggregate. And then what happens next? So what you say, it accumulates, and then eventually it interferes with the cell, and the cell dies off. And you were also talking about prion-like behavior, which many people will know about, but perhaps not everyone knows what prion. So in the UK, prion, we think of mad.
Sure, absolutely. And this is not the same. So although I probably wouldn't eat the brains of, Parkinson's disease patients, but other than that, it's not the same as, Creutzfeldt-Jakob disease. But there is an idea. So absolutely within the neuron, you can get small seeds of fibrillar synuclein. So fibrillar synuclein is kind of the abnormal pathologic form of synuclein. And you can get these small seeds, and it can recruit normal synuclein to kind of build up, and that spreads throughout the neuron. And it's through my research and others that have shown that these aggregates cause dysfunction to the neuron. What's actually fascinating to me is I mean, you do get dopamine neuron death in Parkinson's, but there's areas in the brain where there's no neuron death with large accumulations of alpha-synuclein as well. And that's kind of the main focus of my lab. What is it doing in these brain areas? Is it causing neuronal dysfunction? How is that related to other symptoms of Parkinson's disease, like depression, anxiety, cognitive impairment?
So just so we got it clear, so they basically have this pathology of the alpha-synuclein aggregating in certain cells like the dopamine cells, which we know a lot about. That causes them to die off, and we try and treat that with dopamine drugs. You have other cells outside the dopamine system where alpha-synuclein aggregates, and those cells die off, and they give you some of the non-motor aspects.
So they don't die.
And yeah, and so and then there are other cells where it accumulates, and the cell seems quite happy living with that.
Well, I would not say they're happy. So they're living with it, but our research is really pinpointing that the neurons aren't happy. So they're losing their synapses, and they're not functioning, they're not firing properly. They're not connected with their neighboring neurons.
Okay. So let's
come back. It's not just my research either. Others have shown similar.
But are there cells where it accumulates and the cells seem quite happy? Or would it, if you ever see alpha-synuclein aggregating in a cell, it's bad for the cell?
I think it's bad. I mean, we've shown pretty clearly it's bad for the cell. So one of my graduate students has recently done electrophysiology studies. So it's a way of in the brain. So this is with rodent models, looking in the brains of these rodents, measuring the electrical activity of the neurons with synuclein aggregates, and they are not firing the way a normal neuron did. And this actually happens very early on.
Okay, so obviously POG disease is unique to humans. So how do you model it? How do you model it in the lab? Because it would be really useful, I think, to hear how you model it in the lab, and then how you've studied it to get to this. Because you've already talked about electrophysiology, recording from cells. Presumably you can collect what they're releasing, but how do you model
how do we make these aggregates? So mice, most organisms other than humans do not normally form Lewy pathology. So the way we do it in the lab, and this is what I found when I was in Virginia Lee's lab. So you can take synuclein protein. We actually use bacteria to make large amounts of synuclein protein. We can purify it. You throw it in a test tube and shake it, and it makes fibrils that are very similar to what is in a Parkinson's brain.
Okay.
Now we can take these fibrils, we sonicate them to break them up into really tiny fragments. And I found that you can add them to neurons in a dish, just neurons from a normal mouse, not overexpressing human synuclein or mutant form. We can add them to
healthy young mouse nerve cells. You just
exactly in a dish, and we can make what looks like Lewy pathology in the dish. So then Kelvin Luke at Penn then found that if you take these fibrils and inject them into a mouse brain, we get the same thing. So you get what looks like Lewy pathology. Remarkably, you get a loss of dopamine neurons as well. But you also get what looks like Lewy pathology in the cortex, amygdala, brain regions that are important for cognition, for anxiety.
And do you have to inject it in a certain site, or can you inject it anywhere and it ends up affecting all of the same?
You can inject it anywhere. Brain areas that project to where you, so we can, we have a needle, we can inject it. We inject it in the striatum. Striatum is important in the basal ganglia, which is important,
Yeah, that's where the dopamine ends up in the striatum, isn't it? That's where it's released.
Dopamine ends up in the striatum, but cortex projects to striatum, amygdala projects to the striatum, thalamus. And so all the brain regions that project to the striatum end up with what look like Lewy bodies and Lewy neurites.
Okay, so you inject your sonicated preformed fibrils of alpha-synuclein that you've made.
Right.
And then over a period of time, like sort of weeks or months.
6 weeks, 3 months. Yeah.
Yeah. And then you get pathology in the area where you've injected it and the connected areas.
Exactly.
And that aggregation is just the protein you've injected clumping together?
No, it's the normal synuclein. So this is where you kind of get in. So the prion hypothesis is that you start with a template, a pathological template, and that corrupts the normal protein. Yeah. So what we're looking at is actually the normal protein that's been corrupted by these fibrils that we injected. So and that's actually mostly what we looking at. Yeah.
So the abnormal one, basically, as you say, templates the normal alpha-synuclein. Right. And so then it aggregates. So does the cell, so one of the questions we have is how does that cause a problem in the cell? Is it then the problem in the cell is because the normal alpha-synuclein is now taken out of play and it's put into, so you've got no alpha-synuclein, or is it because the aggregates themselves get in the
I
way?
think both. So initially, very early on, so synuclein is involved in the synaptic vesicle release. There's a lot of great scientists out there that are actually looking at the normal function of synuclein, showing that's involved in synaptic vesicle release. So I think initially when it gets sucked into these abnormal aggregates, you start to get problems with the release of synaptic vesicles, release of neurotransmitters. But then my colleague Hilal Lashuel has shown that as these aggregates build up and they move to the cell body, other organelles in the neuron get stuck. So mitochondria, which are really important for energy in the neuron, are kind of stuck on these Lewy body-like aggregates. Lysosomes, which are important in protein turnover and turning over damaged proteins, are stuck on there. So I think it's both initial, like sucking synuclein away from its normal function, but then this kind of abnormal gain of toxic function as well.
So it sort of gains a momentum. Initially, it's just sort of slightly misbehaving because you can't get enough of the release of the transmitter. And then it starts to recruit more and more, and then that becomes worse. And everything that the cell relies on to keep it healthy eventually gets bumped into these aggregates, and then it
Yeah.
Okay.
Yeah.
And so when you're studying it in the animals, what's the first thing you notice? Do you notice that there's a change in the, you were saying a bit about electrical activity. So is the first thing you notice there are changes in the electrical activity of those cells, or is it the release of the transmitter, or is it that the mouse starts to behave in a funny way?
So the first thing we really notice is changes in the synapses. So changes in the morphology of the synapses, changes in the vesicle release in the synapses and electrical activity. The behavior of the mouse, we don't really notice major changes. You really have to push the mouse, do really complex behavior techniques to really see behavioral changes. So I think the reason for this, we get about 50% dopamine neuron loss. In human, it's 90% loss of dopamine neurons. We can't get more than 50% dopamine neuron loss in the mice. And I think that's the reason we're not really seeing major motor behavior defects.
Why is that, Don? Why can't you get more than 50? If you check
I don't know. It could be something unique to the mice. I actually think it'd be interesting to look at. All of these dopamine neurons are exposed to the fibrils. Even cortical neurons, but not all of them get the pathology. Not all of them are responsive to the pathology. So what's protecting them? I think that'd be, an important scientific question. Why some neurons are resistant?
Yeah, I've always thought that, we always look at the things that go wrong, but actually the
right, but what's going right? Yeah, some neurons are fighting it. So
there are presumably bits of the brain, like the sort of back of the brain, the visual cortex, which is very active because we're seeing all the time, which seems to be pretty resistant to this type of pathology.
It is, but I also wonder, so dementia with Lewy bodies is another disorder related to Parkinson's disease, except that you get cognitive problems first and hallucinations. I do wonder, they're mostly visual hallucinations. I do wonder if the visual system is involved.
So you've got this model where you put in the preformed fibrils, you get this aggregation of the endogenous alpha-synuclein, and that's connected, but some areas are resistant. So one of the questions we have is that's a great model, but is there any evidence that happens in people with Parkinson's? Is there any evidence for this protein spread and then it's seeding pathology?
Sure. So for the spread. So first of all, it is an artificial model. I mean, we are injecting fibrils into the brain, which doesn't happen. So there's a big question, what starts it? What makes synuclein go bad in a human? And so there's ideas that, as we age, our lysosomes, which clear up, damaged proteins, those start to become dysfunctional. So maybe, our lysosomes aren't, clearing up damaged synuclein as well. One of my colleagues at UAB studies environmental toxicants, which can make synuclein go bad. So there's an area there, maybe head trauma. I mean, I really think these questions remain to be resolved. What initiates it? The spread is
that's probably years before we ever see anything, isn't it? So it's quite difficult. Yeah.
Sure. Yeah. Right. And for me, I'm more interested in stopping the spread. So my boss is a neurologist, and he says he sees patients with a tremor in their right hand. They say, okay, I'm fine with this as long as I don't, lose my ability to button up my shirt, my ability to feed myself. Cognitive decline is a major concern. So if we can halt it, and for me, if we can halt it in its tracks, that's really important. So that's kind of what my lab is trying to figure out, how we can halt it in its tracks.
So you think that at the beginning there must be some trigger, possibly environmental, that then
Genetic, environmental,
or genetic in some people, as we
combination, right.
And then the evidence that it spreads in people. So one of the hallmarks often of prion disease itself, like mad cow disease or Creutzfeldt-Jakob, is that in the patients I've seen with it, it starts slowly and then it just absolutely races away with them, because obviously if it spreads from one cell to the another, 1 to 2, 2 to 4, and so on. But that's not the impression you get with Parkinson's. If anything, it comes on and then it sort of slightly plateaus out. So what's the evidence in people that you think this is?
There's a couple ends of evidence. So Jeff Cordover originally showed, he actually did fetal transplants into brains of Parkinson's patients. So they're dopamine neurons. He put them in the striatum, and eventually over time they start to have Lewy bodies. So the idea there was that their neuron, sick neurons are releasing these aggregates and they're being inherited by the new fetal neurons. Then there's pathologic studies from individuals that are deceased that had maybe early symptoms of Parkinson's disease. And so they stage it from early symptoms to individuals that had, cognitive decline and more, longer term Parkinson's disease. So it's based on that. I will say, so in Alzheimer's disease, for example, there are 50% of Alzheimer's patients actually have Lewy bodies. Lewy bodies are pretty restricted in the amygdala, which makes me think that it's not always a spread. For me, my focus is what's going on in the neuron, not as much the spread, but the bad synuclein in the neuron is making the neuron sick. So if we can, prevent that from happening in the healthy neurons, I think that's the way to go.
And do we have ways of measuring it? Can we do this in, how can we measure? I mean, obviously,
so we can measure it in mice easily. In humans, it's there's actually, over the past couple of years, there's been remarkable way of measuring it. So you can't look at a person's brain. Alzheimer's has the advantage that there are PET ligands where you can image, amyloid plaques and neurofibrillary tangles. We don't have that for Parkinson's yet. We don't have a way of imaging with PET ligands, synuclein pathology, but we can take cerebral spinal fluid or skin biopsies, take lysates of that. You have very small fibrils in those, and you add monomeric synuclein or normal synuclein, and it amplifies to get fibrils. So that's been an important advance in being able to diagnose Parkinson's. And about 95% of idiopathic Parkinson's have, it's called a synuclein amplification assay, actually are positive for synuclein fibrils.
So we can take fluid off the back, lumbar puncture, take some fluid, and we can be more confident people have got it. But can that seed assay tell us about whether the protein is spreading in people? So is that mechanism which you had and secondly, can you follow it over time? So do I know people's stage in the condition by using the seed assay to say it changes as we move through the disease?
There are people that were positive with the synuclein amplification assay that did eventually go on to develop Parkinson's. So there is some evidence that if detected early, that was skin biopsies. So,
but you couldn't say I can stage you by just taking a bit of fluid off, say you have this abnormality with your seed assay when you're diagnosed, and 10 years in it looks like this, and 20 years like that.
We don't know. We're still working on, there's research on that for sure. And we also don't know if a treatment that gets rid of these aggregates, we still don't know if the assay is going to be sensitive to reductions in synuclein fibrils, which I also think is important.
So we've established that α-synuclein is critical to the disease. We've now got assays where we can help diagnose people with it. We've also got this idea that α-synuclein, when it starts to go wrong in Parkinson's disease, it affects certain neurons and it may spread and recruit endogenous. I mean, I think it's someone's asked about how much can you spread. So I think it's important to understand this is spread within the brain. So this is not blood transfusions or
no. It's within the brain for sure. And I'll be honest, I question how much it's spread within the brain is what's really going on versus changes in neuronal activity or, when it comes to treatments, I think targeting what's inside the neuron is going to be critical.
Because certainly in the prion field, one of the areas which gave people evidence for it was people having either grafts on the outside of their brain when they
Right,
had
or growth
grafts, or growth
hormones.
hormones, and then
Yeah. And it's not, no,
which we
it's not
never,
that.
which has not been.
That's why I say prion-like. I even hate to
Yeah.
use the word prion.
So I think it's important for people to understand this goes wrong in the brain, and whether it spreads from cell to cell and seeds pathology goes through the brain is an interesting area, but we don't know for certain when that's the case, but it certainly doesn't spread out from one person.
It's not being shed from the body, or right.
Yeah. And so I think it's fair to say everybody who has Parkinson's will have Lewy body if they have.
Well, no. So there are, so there's different mutations linked to Parkinson's disease. So LRRK2 is one of the more common mutations, leucine-rich repeat kinase. There's different mutations. The most common one is G2019S mutation. 63% of those individuals are positive in the synuclein amplification assay, but there's 40% that aren't.
Okay.
And when you have other LRRK2 mutations, maybe only 35% are positive synuclein. So I don't think synuclein is, completely causing Parkinson's. And again, I think there needs to be more research. I wonder if young onset Parkinson's is different than, age 65 and older onset Parkinson's. And what's the role of synuclein in young onset? I don't think we know.
So I suppose what you're saying is there are certain genetic variants which give you a risk of getting Parkinson's, some of which don't develop this pathology, and obviously the very young ones, the Parkin, which are rare, they don't tend to get the psychiatric pathology. So a couple of questions are coming in, Laura, suggesting that you might be wrong. They're suggesting that
I'm not following the chat, so if you don't mind,
that's all right. Leave that with me. People are saying, when we've had all these, because obviously if alpha-synuclein is the cause of the problem, then the simplistic view would be if I give an antibody therapy, an immunization therapy to kill off alpha-synuclein.
Sure.
We'll come back to discuss what form that has to be, and that may be one of the reasons. Then it should stop the disease in its track. But obviously, our very learned audience have been reading up, including a paper from Nature Medicine, I think, which came out last week. About is it when people have been given these immune therapies against alpha-synuclein, they don't seem to have worked.
So there's a few things going on with that. So the reason I've been a little bit cautious about the spread from neuron to neuron is because I don't know how much that is contributing to disease versus what's inside the neuron. So targeting, if we have antibodies that we can know for sure get into the neuron and break up the aggregates or prevent the aggregation, I think that's going to be more important than the neuron to neuron spread. So in the Alzheimer's field, some of the immunotherapies seem to be working. Amyloid plaques are definitely outside the neuron. And so it's a different mechanism than with Lewy pathology. So there's that. The other thing is we have no way of detecting if this immunotherapy is even doing what it's supposed to be doing. So again, in the Alzheimer's field, we have PET imaging for amyloid beta. And so we can know that the antibodies are actually clearing the amyloid beta from the brain. We don't know that in the Parkinson's trials at all. That's why I think the models are important because, and people have not been doing this, showing that these immunotherapies actually work in the models. And so we don't know if they're actually working like they're supposed to be. So there's a lot going on.
So in your models, have you tried these antibody therapies to see if they
I have not.
But have other people done that and shown they've worked, or have they?
So my former mentor, Virginia Lee, they have tried it and it did work. It was high levels of antibody in those. There's one, I think a Biogen antibody that went to clinical trials and failed. And the data is buried in the supplemental figures. It didn't work. It didn't clear the aggregates in the mouse model. So
But your basic message is the reason these trials have failed might be not because the hypothesis is wrong. It's just that you've given the antibody therapy, it hasn't got into the brain, it hasn't got into the right area. And I suppose one of the problems I've always wondered is I'm not quite sure what they're targeting. So which bit of alpha-synuclein are these? Is
it normal synuclein? Is it abnormal synuclein? I'm always concerned too, targeting normal synuclein. We don't know yet if that's going to be good or bad. So, there's also the antisense oligonucleotides, so that reduces levels of synuclein.
So that's
And
a
that's
way of trying to
producing,
stop the gene from coding for it, if you like. It sort of interferes with that, so you produce less of the alpha-synuclein rather than getting rid of it.
exactly. So you're producing less of synuclein inside the cell.
Yeah.
And it's nice because you're not really getting rid of synuclein, which I wouldn't suggest. You know, you can just lower it a little bit, and that may help stop the progression. The problem with the antisense oligonucleotides is it requires spinal cord injections, intrathecal injections, probably every 6 months. Yeah, I don't know a lot of people that really want to sign up for that. Again, I think if you can develop some kind of pill that you could take, maybe to lower synuclein in your brain, you know, that's in the future.
Yeah, I mean, I agree with that. I mean, I think, you know, obviously the more convenient way you can deliver things, the better. We've done trials,
Absolutely.
not in Parkinson's, in Huntington's, where we've had to do injections into the spinal fluid on a frequent basis, and it's not something people queue up for unless
No.
they know it's going to really make a major difference.
Sure.
So one of the questions, so obviously we you know, the reasons the trials may have failed is for the reasons we've said. You're not quite sure where you're targeting. Is it getting into the brain? Is it getting to the right compartment? Obviously, in the world of Alzheimer's disease, and this is one of the questions, or a couple of questions coming up, it's about when you go. So, you know, Alzheimer's disease, obviously the original trials in people with well-established disease. Now there's this great push to try to pick people up before they even know they've got it.
Right.
And do you think this is the direction of travel for your
I was just
targeting
talking about
alpha-synuclein?
that. So I think about my family a lot. My family will not go to the doctor unless they know something's wrong. So I feel like, you know, my family won't go to the doctor unless they have a tremor. So, and that by that point, you do have low pathology in your brain. Probably, you know, 80 to 90% dopamine neurons are gone. So for me, I feel like really the focus should be on progression.
Right.
Eventually in the future, say I you know, get a blood test, and my blood test says I have high A1C, high cholesterol, and high levels of synuclein. And you might want to start thinking about taking a statin and, you know, this drug to prevent synuclein aggregation. I mean, that's way in the future. But I can tell you, you know, if someone said I had a 20% risk of getting Parkinson's, you know, can we inject this AAV virus in your brain? I'm not going to sign up for that. So you know, again, I feel like for me, it's the progression, you know, preventing it from getting worse is kind of my focus.
Because it's always a difficult one, isn't it? Because you think, well, should we go screening for everybody? And then you think, if you screen and you can't do anything about it, is that a good thing to do?
Right.
But obviously, if you don't start screening, then you find something, the therapy, then you haven't got a population to test. So I mean, we've obviously established a lot about alpha-synuclein. So where is your work at the moment, Laura? So what's your main research focus at the moment, and where do you see that developing, and how will some of the therapeutic interventions we talked about?
Sure. So I have kind of two main areas in the lab. One is using the fibril model to really understand what happens very early on with the synuclein aggregates. I feel like, you know, if you're starting to get synapse degeneration, for example, that's something that we can intervene. So once the neurons die, I think it's a little bit harder to intervene. But if you can somehow improve firing of synapses, you know, that's something that's really interesting to me. And also, you know, what's causing the disease, you know, at a circuit level, at a synapse level. So that's one area.
So can I just pick up on that? So is the strategy, obviously, you know, you put in your preformed fibrils, you look very early, you see that the nerve cells have a subtle defect, which you know where it's going. So what's your sort of, so you understand that pathway, but then do you try and interfere with it? Or because you could say, well, actually, I'm not really bothered about that. If I just got rid of the alpha-synuclein, how it does it. So have you got clever strategies where you're trying to think, we could recruit how the cell can help itself here?
They're not my clever strategies, but there's all these omics studies which are kind of looking at you know, what proteins change in a Parkinson's brain or in this fibril model, and a lot of synaptic proteins are coming up in that. So I have a colleague, Thomas Beater, who studies this protein called orexin, which is really important in neuronal communication. And he's showing that when you overexpress orexin, it can actually prevent the spread of the synuclein pathology, but also improve synapse morphology and firing. Again, that's probably, you know, manipulating orexins. It's probably not going to be a treatment. But I do feel like if we can get a better understanding what's going on with the biology,
But I guess the strategy is that you know that this synuclein causes a problem, and I don't need to go after synuclein. I can work out other mechanisms that can help compensate for the problem.
Exactly.
Yeah.
Exactly. Right.
So that's one area. Where's your second major area of?
My second major, there's a gene GBA1, which again is one of the more common gene mutations linked to Parkinson's. Individuals with GBA1 mutations have a 5-fold risk for cognitive decline.
Yeah.
So my lab is really interested in so GBA is a protein in lysosomes. Why are defects in GBA1 in the protein leading to cognitive decline? And my graduate student, Casey Mahoney, has found using mice that have mutant GBA on one gene, a normal GBA on another gene. She finds these mice have major spatial memory defects alone. So even without synuclein pathology, there's something wrong with these mice. And she's actually finding that it's a particular subset of neurons that are impaired in these mice. So we're wondering how dysfunction in lysosomes, in certain lipids that are in these lysosomes, how that can be kind of priming for cognitive decline in Parkinson's disease.
So let's just slightly unpack. So GBA, this is the Gaucher's gene. So
right.
So if you get one abnormal form of it, increases your chance of getting Parkinson's. And then people who get two, they get Gaucher's disease, of which.
And Ellen Sidransky just won the breakthrough prize for that.
Yeah, for making that observation.
Yeah,
and then that gene defect is not only predisposes you, if I've understood this correctly, for getting Parkinson's, but it also increases your chances of getting a more aggressive form of Parkinson's, which has cognitive problems.
Right.
And you're saying that this GBA, and this is localized to the lysosome, which is a bit of the cell that breaks things down. It's your sort of dustbin car, like sort of gets rid of things. In itself, GBA1 mutations makes your cells more vulnerable to disease, if I've understood it correctly.
That's
and then
what
if you throw in
Casey is
alpha-synuclein as well, it's a sort of double hit and you're in serious trouble.
Right. And that's what Casey is finding with that.
Okay. And so when you say, the cognitive problems, is this cortical? Is this all, basically problems in the cortex, or is it, hippocampus, which is very much associated with, navigational memory and Alzheimer's? Is it selective for areas? Because you've already talked about this, that these diseases, whilst they're distributed, they're not everywhere.
So Casey is finding it's entorhinal cortex, which does project to the hippocampus and is involved in spatial memory. But we have not, this is all new, so we have not looked at other areas of the cortex. And, it may also be not just neurons, but microglia, which are important for responding to damage. And so, there's like a whole wide open area of research now.
So what do you think is, so you've talked about what you're really after is trying to slow down the disease, go after what's driving it. So what do you think are the key factors that drive the progression of Parkinson's? Obviously time, but there's not much we can do about that. GBA,
time, aging. I really think lysosome dysfunction. Because that happens normally with aging. Casey also found there's this buildup of a particular lipid that actually just happens with aging in this mice, and there could be something with that lipid that's causing.
This is a lipid in a membrane, so lipids are normally found in membranes, like cholesterol and things like this.
Right.
So
and one particular lipid is building up. Yeah.
Okay. So there's a problem in the lipids, which then interacts with the lysosome, which then could interact with GBA activity, which is within the lysosome, I guess, driving activity. And then you've also got alpha-synuclein. So it's a sort of multi-hit.
Right.
And so if you're going to target this, so obviously different people progress at different rates. So how can people take this piece of information and sort of use it to stratify patients or to better treat them? Or is there anything we can take from these models that might enable us to do things in the clinic in a slightly smarter fashion? So
with the GBA mutations, my colleague Roy Alcalay has found that patients, some Parkinson's patients have high levels of this lipid glucosylsphingosine in their plasma. So that could maybe be used as a biomarker. One of my graduate students, Ariel Manabot, is looking at drugs that reduce this lipid. Hopefully it'll work. We're not even near understanding that yet, but she's working very hard on that.
So is this a normal lipid, Laura, or is this an abnormal? Is this a normal lipid or an abnormal?
It's a normal lipid, but it gets higher with aging and with GBA mutations.
Okay. So why does that happen? Why does it get higher when we get older?
I don't know. I don't know.
Lots of things get down when we get older.
Yeah. Right.
Okay. But it does go up, and this is something you can measure in the blood as well as in the brain, or is it just something you can
in the plasma? We can measure it in the plasma, so which is nice.
And so, I mean, obviously, we've done some work ourselves on Parkinson. I mean, age is a big determinant of where people do. So, I often say to people in the clinic, if you've got a young onset, the bad news is you have many years ahead of you. The good news is, the cognitive problems are much less of a problem when you're younger.
Right. And I think young onset may be very different. And I don't have a lot of evidence to point to for that, but I do wonder, if when you're 65 and older, you're going to have the lysosome problems, likely Lewy pathology. With young onset, I'm not sure. So I did see a study recently on exercise, really improving abundance of dopamine transporters.
Tai Chi is a big thing. There's several trials of Tai Chi showing it. I don't quite know what Tai Chi does for you.
Or high interval training, HIIT training. So there's a lot of interesting research going on that, but that doesn't have to do with synuclein.
And so just trying to wrap this up in terms of, so your hypothesis would be that α-synuclein is clearly an integral part of the disease process. Quite how is yet to be fully elucidated.
Right.
But your view would be that the cognitive problems which you see aren't actually so much related to faster protein spread, but actually just widespread pathology, which may begin in the dopamine system, in this entorhinal cortex, in the hippocampus, in the cortex. So when cognitive problems occur, it's because basically lots of networks start packing up together rather than it's one packs up and then it passes its problem to another. That may occur, but it's not what your hypothesis would be. It's multiple areas going down.
Lysosomes are defective, synaptic vesicles start to become defected, the synapses start degenerating. And I think, it's a combination of maybe lysosomes, synuclein, mitochondria.
So coming back to where we began, why do you think it's only certain areas that
so some brain areas express high levels of synuclein, whereas others don't.
So you think it's related to the fact that your networks in the brain have an underlying vulnerability because they have higher levels of this, and presumably, and now I'm completely speculating like I do most of my life, but I mean, presumably, you have high levels because they're quite active because it's involved with transmitter release.
Sure.
And when in the good old, when you're running around the Serengeti plain, you weren't expected to get beyond 50, and so having lots of activity in your nerve cells was a good thing to keep you alert and lively. And once you got to 50, you'd probably be dead from something else, so it didn't matter.
Right, exactly. Yeah, and over time, you get a buildup of reactive oxygen species, damaged mitochondria. It all kind of
Okay. And so, sort of finally, one of the areas, we've been talking about aging. So, I mean, obviously, you're giving a wonderful, seminar, if you like, on alpha-synuclein, its links to Parkinson's and how it may be causing problems. One of the things which you said, which caught my attention, which is also something which is of interest, I think, is with aging, you get other pathologies. So you were saying, most people have a bit of Alzheimer's, amyloid pathology. So how much do you think these pathologies talk to each other? So, one of the questions which comes back to these trials, I've always said, is, you treat people with alpha-synuclein antibodies on the assumption that the disease is an alpha-synucleinopathy. That's all it is. But from what you're saying, as you get a bit older, you get a bit of Alzheimer's pathology, you get a bit of vascular pathology, small blood vessels close off. So how much do you think these pathologies feed off each other and help drive the disease? Because the old theory used to be, a lot of people get cognitive problems, Parkinson's, because they actually get Alzheimer's disease, not necessarily Parkinson's dementia. So what's your take on that?
I know with the Alzheimer's patients that have Lewy pathology, their progression was a lot more rapid to the cognitive decline. And that's mostly synuclein in the amygdala. And I think, people with multiple pathologies and Parkinson's. So we were looking at dementia with Lewy body brain, all of the patients that we've looked at have amyloid plaques, neurofibrillary tangles, and Lewy pathology. So I think it can add up. And again, this is why I'm getting back to like the lysosomes, because why are all these proteins building up? So maybe all these proteins, there's high levels of these proteins in the cell. They're kind of in this disordered conformation. So maybe there's something wrong with the clearance of these proteins, and they're just building up and causing problems.
But you would see it that there's a sort of problem in this. So, the simple analogy would be the dustman's not coming every week, coming every 2 weeks, and your house is just filling up with junk and you're not getting rid of it. So it's a bit agnostic to what you're collecting, whether it be alpha-synuclein or amyloid or whatever.
It's just a hypothesis, right?
Yeah. Or do you think that once you start to get a bit of pathology, say with tau, which is part of the Alzheimer's, that makes you more likely to get alpha-synuclein pathology? So one pathology increases your risk of another, which is that they're sort of seeding each other rather than there's just a sort of problem with the dustbin man. If you started collecting one, you're going to collect the other.
So we did some we haven't published this yet. We've looked at these dementia with Lewy bodies brains, and the tau and synuclein are in different cells. Okay. So I don't know if they are cross-seeding each other.
Okay.
Yeah.
So you don't think there's an interaction at the protein level? You think there's a problem with the sort of cellular machinery?
I think so.
Whatever protein is going to cause a problem will accumulate in those cells because of that.
I also think, Spanish flu. So back in 1917, a lot of people, the Spanish flu, and a lot of those people ended up with Parkinsonism. They all had neurofibrillary tangles and not Lewy pathology. And a lot of the LRRK2 mutant patients have neurofibrillary tangles. So, yeah.
So looking to the future, where do you think the answers lie? So Jodie's written in saying, we've known about alpha-synuclein for decades. Why is it so difficult to get to the bottom of this? I mean, I think you've answered that question to some extent in that, it makes up a lot of our brain. It's got complex functions. So just trying to rip out alpha-synuclein is probably not going to be a good thing.
And we haven't had strategies to just rip out synuclein either until more recently. So
where do you see, what would you see in the future as how we're going to, would it be a combination of therapies which sort of deals with autophagy, so this lysosomal function, a bit of antisense, knocking down a bit of alpha-synuclein, trying to give some of these other, trying to target other pathways to induce plasticity? How do you see the sort of therapeutics developing out of this?
I would think some combination therapies. Identifying who has synuclein pathology, I think, would be the first step. I don't think it's, there are people with Parkinson's that don't have synuclein pathology, so I don't think synuclein therapy would work for them. So if we can find the subset that does have synuclein pathology, trying to reduce abnormal synuclein in the neurons, I think would be a good step there. Maybe boosting lysosome function. Something else I didn't mention is that their microglia and some of these inflammatory cells are also releasing cytokines that are especially killing the neurons and making them sick.
So you're much more of a sort of combined.
Possibly. Yeah, I know that's hard to do. People kind of push back on me, like we'll never be able to have a combined therapies.
Well, I'm a great fan of that.
You are? Okay.
Yeah, because I think unless you know exactly what you're doing, so if I think of medicine over my 40 years I've done it, some of the most successful therapies have been things like HIV, which is combination therapy, anti-TB medication, heart disease. These are combination therapies which work very well together.
Right. I even think of like cystic fibrosis. The combination therapies are actually curing the disease.
Yeah, well, cystic fibrosis is a great disease to talk about because it was the patient community that solved the problem by giving the funding.
And it's the ones, bench to bedside, figuring out what's going on biologically, getting the right treatments, and then they actually found a really good animal model. I think some of our problems have been not the right preclinical models, and then getting it into actually almost curing the disease.
Yeah. So. Well, that was one of the comments actually that people, I can't remember who mentioned it, was about, one of the problems with the animal models is quite often, you give the therapy, then you give the lesion, and then you show it doesn't develop, which of course is not what happens in real life. It's you have to have the disease going before you can then try and stop it.
So I just saw a comment that combined therapies will never get funded. And so this is completely out of my area of expertise. And I've heard other people say the same thing. So I don't know why. I guess because you
Depends,
can't
I think, what
really,
you combine. So I think if you combine drugs which are already have clinical,
I
have
see.
repurposed, then there's no reason you couldn't do it. I mean, I was at a meeting this morning where they're doing metformin and clemestine, which is an antihistamine for MS, as a way of remyelinating the brain. So you can do combination therapies that are out there. Obviously, if they're novel therapies, then you'd have to do one.
I see. Okay. Because you have to know each one separately, what's the
Yeah, and of course, if I gave
toxicology.
you, Laura, 2 experimental drugs and you went completely crazy, I wouldn't know which one of those 2 drugs would make you
Right,
completely crazy.
right.
So I mean, we're coming to an end now, but I just want to thank you for taking us through this story. I mean, a lot of people won't know just how seminal you've been in the work of this. And 15 years ago, there was no such thing as a preformed fibril model of Parkinson's disease, and we relied on toxins and transgenic, where you make mice with abnormal genes. So this work has been transformative, and I think identifying what goes wrong in those early stages and how alpha-synuclein may be triggering the disease and then driving the disease, and putting that in the context of all the other problems you've mentioned is so important, really. So
Thank you. Yeah.
So it's been
Hopefully-
very inspirational. I think you've also explained the alpha-synuclein trials, which I know a lot of people get quite worked up about. That if a trial doesn't work, it must be because the therapy doesn't work, rather than you haven't got other things.
We have no way of knowing it's working. We need target engagement for
Yeah.
these.
Which is a very good point, of course.
Which it may, the immunotherapy may not be working, but we don't know.
Yeah.
Without
Yeah.
knowing if it's clearing synuclein. So,
And you make a very good point. What we do know about amyloid immune therapy is that the amyloid disappears in the brain.
right, we know that.
Post-mortem. So we know that, whereas we've never, I don't think in the alpha-synuclein immunization anyone's died and they've looked at their brains. I don't know that, but that would be
Right.
Yeah.
Even the amyloid immunotherapy, it is working, and it's really exciting because there is some progress finally in neurodegenerative disease, which we never had before. But it's still not, 100%. And people are looking at reducing tau. Because the idea there is maybe amyloid is initiating tau aggregation.
No, I mean, I find it quite exciting. I mean, I can be a bit cynical about it, because by chance 1 in 20 trials will work. And so if you've done 40 trials, which they have done now, Alzheimer's, you'd expect 2 of them to work.
No, that's fair. Yeah.
They've now got 3, so they get that. And I always remember somebody presenting a talk where he showed, as a pathologist here, showed all of the people on the very first amyloid immunization therapy had no amyloid in their brain, but they certainly didn't have normal cognition. So
Right.
I think there are important aspects which we learn about, and it gives us hope, but we do need to think a bit more about how we can better do it. And I think you've given us great hope there, Laura, because your work and those of your colleagues is fantastic about how you set about modeling it and understanding it and then translating it. So I want to thank you again. Fantastic talk, a fantastic audience, tremendous number of questions, I have to say. So I'm seriously impressed by the number of questions and the level of them. They're absolutely
I'd like to get at all the questions so I can think about.
Yeah, because then you'll see all the hidden all the nice ones from you, Nora. But
well, and then I want to, and push back. I mean, it helps me think.
No, they've been terrific, and you've been terrific. So thank you very much
for, great.
for your time.